This is part 2 of 7.06, and I added to our notes polypeptides bend or coil because of hydrogen bonds between nearby amino acids. This makes the polypeptide protein have a certain shape. Shape is very important for reactions and function. And I just kind of put in this picture. If you notice, it's like really messy. But you can see here how they have boxed out the different amino acids, or in this case, the different chemicals, and they said, see how this oxygen is partially negative and this hydrogen is partially positive, so they're going to want to be near each other. And the same thing over here. This oxygen is going to want to be near this hydrogen. And you can really see then how you get the bends and the folds created. And then I also added farther up into our definition of a protein, there it is, molecule made of one or more polypeptide chains that has a specific function in a living thing. Alright, so this is showing protein synthesis, or how you make a protein. You have the primary protein structure. Amino acids linked together through peptide bonds form long polymers called polypeptides. So again, the amino acid would be the monomer. You put a bunch together, you get a polymer. You put a bunch of peptides together, you get a polypeptide. So again, you can see the nitrogens are connected to the carbon double bonded oxygen group. You can see that pattern. All of these blocks represent the R groups, those different functional groups, to show that this is a different amino acid than this one, which is a different amino acid than that one, which is a different one than this one. You can also have secondary protein structure. Amino acids linked together through peptide bonds form long polymers called polypeptides. Then you have possible polypeptide structures. And you can see here you have hydrogen bonds. So again, if this one ends in an oxygen and this one ends in a hydrogen, they're going to want to be together because negative, positive attract. Tertiary protein structure. Amino acids linked together through peptide bonds form long polymers called polypeptides. So they're huge. And quaternary protein structure. Amino acids linked together through peptide bonds form long polymers called polypeptides. You can have protein 1 linked together with protein 2. And those are your different levels of proteins. Alright, so we talked about proteins. Now we're moving on to the other one, which is nucleic acids. So nucleic acid consists of nucleotides. Another biologically important class of organic molecules is nucleic acids. Nucleic acids consist of deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. So if you notice, the only difference is the deoxy. These molecules carry the genetic information that determine traits such as hair color and disease resistance. Nucleic acid are polymers of nucleotides, and a nucleotide is specifically a subunit of nucleic acid that consists of a phosphate group, a 5-carbon sugar, and a nitrogenous base. So for nucleic acids, nucleotides are the building blocks. Just like amino acids are the building blocks for what? For proteins. Alright, so each nucleotide consists of a sugar, and that specifically that sugar is ribose in RNA and deoxyribose, remember ose for sugar, in DNA. It also has a phosphate attached to the sugar and a nitrogenous or nitrogen base. There are 2 classes of nitrogenous bases. There are purines, which are 2 ring structures, so adenine and guanine, or A and G, and pyrimidines, single ring structures, so cytosine, thymine in DNA only, uracil in RNA only. So if you remember from biology class, you have the A, G, C, T, or U. This is why they're different. It has to do with the chemistry of them. Nucleotide geometry. DNA's unique structure is formed by linking large numbers of individual nucleotides. When the 2 nucleotide strands of DNA molecule are untwisted, they resemble a ladder in shape. A DNA molecule consists of 2 strands of individual nucleotides linked together and twisted to form a double helix. The nucleotide group consists of a sugar, a phosphate group, and a nitrogenous base. And then they're going to link together, of course. Just like the amino acids link together to form a protein, the nucleotides link together in order to form nucleic acid. So let's add some stuff to our notes. So I added to our notes, nucleic acids, DNA and RNA, are made from nucleotides. Nucleotides are made of sugar, a phosphate attached to the sugar, a nitrogenous base. And the nitrogen bases, you can have the purines or the pyrimidines. Nucleotides link to form nucleic acids. Like proteins and other organic monomers, nucleotides link together by a condensation reaction. For nucleotides, the reaction involves the phosphate group on a carbon of a sugar of one nucleotide and the OH group on a carbon of the sugar of an adjacent nucleotide. The OH group from the one carbon and the H from the OH portion of the phosphate group are removed as water. The remaining phosphate oxygen atom and a carbon form a phosphodiester. Okay, so phosphorus, di, 2, and then ester. A phosphodiester bond that links the two nucleotides. Similar reactions occur to form chains of nucleotides, which results in DNA or RNA. So they come together, boom, you get a water. Okay, so let's look at that again. Oops, didn't stop it soon enough. All right, so we have the OH here, and it comes over there, and that's when you get the water, and then it simply connects here, right here, where they are bonded together. That is your phosphodiester bond. All right, so basically this oxygen and this phosphorus are what's going to come together here. That's this oxygen and this phosphorus. And then these two, you can see that this is an H and an OH. Those are what's going to pop off as the water. So the H and the OH, and then this oxygen is going to attach to the phosphorus right here, and then this is going to be your phosphodiester bond. So the phosphodiester bond is again between the phosphorus and this oxygen. So that would be your phosphodiester bond. And then they just keep doing that, and then of course that is what makes the long nucleotides coming together is going to be your DNA or your RNA. I also want to take another look at the picture. You can see the similarity here. They both have the phosphorus in those groups here. Start over, there you go. They both have, you can see that this is the same in both. They both have a sugar. And you can see this one has one ring and this one has two rings. And if we back up, we can see if it has two rings, it's either adenine or guanine. If it just has the single ring, it's either C, T, or U, cytosine, thymine, or uracil. So it's good to see that, oh yeah, it's got that same phosphorus going on, the same sugar, and then it's just the difference of the one or two rings for if it is a purine or a pyrimidine. DNA is a double helix. DNA is the primary genetic material in your cells and those of most other organisms. Its structure was described by James Watson and Crick in 1953. And it's important to know that while they described it, they did not do the work to actually find it. And so there's been huge debate over who really deserves the credit, including Rosalind Franklin, who actually died because she did all that x-ray work. Because when you're working with x-rays, you know, when you go to the doctor or the dentist, they always put that lead vest over you because it actually messes with your cell's DNA. And she died from radiation in that. And so it was her work that was very important for them figuring out and putting together the structure. So two strands of nucleotides are twisted about each other in the form of a spiral staircase called a double helix. The sugars and phosphate bonds form the backbone of the helix. The nitrogenous bases face inward and are bound to each other like rungs on a ladder. Hydrogen bonds between purines and pyrimidines hold the double helix together. So again, the hydrogen bonds is when you have that hydrogen that's partially positive, and usually it's attracted to an oxygen, and it's because the oxygen is partially negative. Adenine binds with thymine, and cytosine bonds with guanine. The structure can unzip to replicate. Each side serves as a template for another. Information in DNA specifies the amino acid sequence in proteins, so they're related. So strands of DNA after an extraction appear in a test tube. So if you were to get some DNA from your cheek cells, this is what it would look like. And it kind of feels like snot. It's very interesting. And this is a complex model of the double helix structure of DNA. So this is all stuff from biology. Definitely, if you're like, oh my gosh, I didn't remember any of this, add it to your notes. Proteins and nucleic acids are important polymers. Amino acids consist of essential carbon bound to a hydrogen group, NH2 group, COOH group, and a variable R group. Amino acids link together through peptide bonds to form long chains, which twist and fold to form proteins. The particular sequence of amino acids determine the protein's shape and function. Nucleotides are monomers for nucleic acids. Nucleotides consist of a sugar, a phosphate group, and a nitrogenous base, either the purine or pyrimidine. Nucleotides link together through phosphodiester bonds to form nucleic acids, DNA and RNA, which carry genetic information. This would be an excellent paragraph to copy, paste, put in your notes, and then all these important words like this, put a line, and then tomorrow go back through. Can you fill in all the blanks? Organic molecules, example, nucleotides form nucleic acids. Organic molecules, amino acids, form proteins through peptide bonds.